So first of all, I would like to thank the UCLA group for inviting me, and this is my first time attending the CGSI, and I really enjoyed it, and thank you. And so this is a tutorial, so I will give an overview on analyzing the large-scale multi-ethnic whole genome sequencing studies. So in the last 15 to 20 years, the field has switched significantly from the GWAS data to the whole genome sequencing data. And about 15 years ago, that was the launch of the 1,000 genome project. At that time, the sequencing cost was quite high, and so therefore only about 1,000 people were sequenced, and the depth was pretty low. And so in the last 5 years, the sequencing cost has dropped dramatically. And in US, and there were 2 large whole genome sequencing programs. One is called TopMed, is called Trans-Omics Precision Medicine Program by NHGRI, and it has sequenced about 190,000 samples. And the other is called a Genome Sequencing Program by NHGRI. It has sequenced 140,000 whole genome, 220,000 whole exome. And so we are likely to make those data available to the community later this year. And and also several large biobanks have launched the genome sequencing effort. And for example, UK Biobank and yesterday just released the 450,000 whole exome, and also 200,000 whole genome. And then Million Veteran Program plan to sequence 1 million people, and also all of us plan to sequence 1 million people as well. And NCI is launching its own sequencing program, and also the unique aspect of that is provide the somatic tumor sequencing. And so this gives you an idea about how the TopMed data look like. And for Freeze-10, and the TopMed has sequenced about 190,000 whole genome, and with deep sequencing 38x, and it's multi-ethnic. Both the CCDG and the TopMed are multi-ethnic, and then you can see it has very good distribution across different ethnicities. And also TopMed has the sequencing samples have many phenotypes, including the long, hard, and multi-phenotype, and blood and sleep. And also part of the TopMed sample has a multi-omic data available. And so in TopMed 2, and they will have the single-cell sequencing data available as well. And so what are the major features of whole genome sequencing and the biobank data? And first is the massive real variance across the genome in the whole genome sequencing data. And over 97% are non-coding variants. For example, in TopMed Freeze-10, if you look at a figure here, and so you can see before the QCs, totally there are about over 1 billion variants, and which have been measured and observed, and among them about 850 million variants are past variants, and among them about 45% are singletons. So that means only one person has that variance in the TopMed sample, and about 15% are doubletons, and so common variants only occupy less than 1% of the total variance, and also about 70 million are indels. And so this presents substantial challenges when we deal with the whole genome sequencing data, because a massive number of variants and also massive number of real variants. So given a student may not be familiar with the whole genome sequencing data and the challenges, I'll start from the common variants. And so in the GWAS data, basically it's array technology. So if you look at this toy example, the top 3 people are, say, lung cancer cases, and the bottom 4 people think about the controls. And so because the GWAS array technology only focus on the common variant, so therefore it only genotype this particular loci. And so if you look at this loci has 4 T's and 3 C's. But if you look at this particular location, this loci here, you can see there's only 1 A, all the rest are G's, and those variants are too rare. And so they are not measured in GWAS. And so because GWAS only collects the common variants, so therefore the traditional strategy is to do a single SNP analysis for association. Why we do sequencing? The reason is the GWAS data only genotype the common variants, which occupy less than 1% of the genome. And so the sequencing basically genotype every single base pair across the genome. And so therefore we'll be able to get a whole genetics code for each person. And so because the majority of the variants across the genome are rare variants, so therefore the analysis strategy has to be changed. And because the variants are rare, one cannot analyze one SNP at a time. So the strategy is to analyze a SNP set at a time. So this is where the challenge and art is. How can we define the SNP set for both coding and non-coding regions? And so there are 2 major features of the large biobank and whole genome sequencing study. The first is multi-ethnic. And so both the GSP and the TopMed and really make a significant effort on addressing the health disparity in this genetic research. So if you look at the TopMed disparity, the ethnicity distribution, you can see that very good representation of different ethnicities. And also Million Veteran Program, you can see about 72% are Europeans, 12% African American. And the second major feature in both the GSP and TopMed and the biobank data are the samples are related. And so if you look at the figure on the left, and then you can see that about 1/3 of the sample in UK biobanks are related samples. And also if you look at the figure on the right, and you can see the large number of samples in TopMed are related sample as well. So therefore we need to address both population structure and the relatedness and in the analysis. And so this talk has two parts. So we focus on how can we do a scalable analysis of a large whole genome sequencing and biobank data. The first part is on the real variance association analysis. So I'll give an overview about the methodology, in particular like Bowden, SCAD, and also the STAR method. And then the second part is on meta-analysis. We can see how much time we have, and then we'll talk about the meta-analysis. So let me talk about the first part. And so how can we perform real variance association analysis in whole genome sequencing study? And so in particular, how can we incorporate functional annotation? So this is where the genetics and the genomics meet. And so we need expertise of both fields. So let's start from two examples. Suppose the phenotype is LDL, and then suppose we have sequencing data, and suppose this region is the ApoE enhancers, and suppose there are 100 variants in the enhancer ApoE of enhancers, and because majority of the variants in these regions will be rare variants, so therefore the traditional way do the single SNP analysis in GWAS does not work. Then we need to perform the SNP set analysis. So that is where the challenge is. How can we define the SNP set? And so here in this 2 example I give you is like, suppose we define the enhancer. This is a SNP set. Those are gene-centric analysis. Then how can we define the enhancers? And so this is quite a challenging task because this is a non-coding regions. And so this will be the cell and tissue specific. And also within this region, suppose there are 100 variants and only a subset of the variants are likely to be associated with the phenotype. And so this is just to give you 2 examples here. So suppose only 4 variants associated with LDL, all the rest are nulls. Then how can we perform the test? Then the null hypothesis will be we test the beta. This will be the 100 beta, and because each beta associated with 1 SNP. And then we test the null hypothesis. There's no association between the enhancers ApoE and the LDL. And then we need to adjust for the population structures and such as the PCs, and also the relatedness. We can use random effect. And so we don't want to do 100-degree freedom test, and that will lose too much power. Then, so what is the main idea of the relevance association test? So basically the idea is we need to aggregate individual association test statistics within a SNP set. And then that is where the art is. How can we aggregate them powerfully? And so that is how can we integrate the omic data and also the whole genome sequencing data. So if one goes from the variance to function to phenotype, and the first is we want to borrow information, functional information from the omic data that can help us to empower a real-variance association test. So in particular, if the variants are more functional, have more regulatory function, we want to upweight them more. If the variants have more conserved, we want to upweight them more. And so this is the first part, how we can use the omic data to help us with the real-variance association analysis. The second part is that we want to identify the causal pathway. And from the genetic association and to identify the causal pathway that's this far. And so therefore, we want to incorporate functional data to help us how the variance of, what is the mechanism of the variants affect the phenotype, understand the causal pathway. And so this can be done through the causal inference, such as a causal mediation analysis. And for, then after we do that, then that is where the action will come in, and then that can help us to understand the potential prevention intervention strategies and also identify the drug target. And for today's talk, we are going to focus on the first part. So how can we incorporate the functional information to help us empower real-variance association test? And so to this end, the first thing we need to do is to functionally annotate any whole genome or whole exome sequencing study. So GSP has the functional annotation working group. And so the goal of this working group is to integrate the data from many different sources to construct a functional database that can be used to annotate any whole genome sequencing study. And so we call the favor. And so this includes the functional informations and from the ENCODE roadmap and CAD, GTAG, and many others. And so then right now there are over 300 annotations and have been incorporated. 80% of them were constructed using build 38. And those annotations include the allele frequencies, such as ethnicity-specific allele frequency, conservation, protein function, epigenetic score, mapabilities, and the ClinVars and EQTL information. And so because any whole genome sequencing study, when you increase the sample size, and then you are going to observe more variants. And so therefore, in order to annotate any whole genome or whole exome sequencing study, we have annotated all possible variants that include 3 billion positions, 9 billion variants. And so the favor database right now includes annotation of all 9 billion variants. And so this, you can visit this website, and I'll do a little demo in a minute. And so this can allow for single variance annotation and also allow for region annotation and also gene-based annotation or batch annotation. So what are the use cases? And so there are multiple use cases. First, after the GWAS studies, suppose you identify the top variants, and then many of the top variants are likely to be in LDs. And so one need to decide which one want to follow up for functional experiment. And so this can, using the favor database, one can help prioritize the GWAS hit. and also can help with the fan mapping by incorporating functional information, and also can help with the individual variance checkup and to see which variants you want to follow through with your experiments to identify whether they are causal variants from the GWAS hit, and also can identify the functional variants in the gene or regions, and also can annotate any large whole genome or GWAS studies such as UK Biobank, and also can facilitate real variance association studies. And so now, suppose I have like a top metadata or UK Biobank whole genome or whole exome sequencing data. The first thing that I need to do is annotate the UK Biobank whole genome sequencing data. And now we have this backend database and Favor, which has annotation of all 9 billion variants. And so it is currently in the cloud, and so one can download the whole database. And now, suppose you have raw VCF files, and so the first thing you need to do is to QC the data, and one can use a Hill to QC the data. After you QC the data, and now suppose how can we annotate the UK Biobank data and or the GSP or TopMed data? So that is how we use this Favor annotator, which is our program, and that basically call the Favor backend database and then annotate the TopMed data. For example, TopMed data has 1 billion variants, and then in this database has 9 billion variants, and so basically we annotate those observed variants in TopMed. And so then that will create annotated whole genome sequencing data, and so we save it in AGDS format because the compression rate is much, much smaller, and it is almost 1,000 times smaller compared to VCF file. And now one can perform the analysis and using we call the STAR pipeline, and using the STAR pipeline to perform the real variance association analysis in the whole genome sequencing study. And to do this, one can either do this using in your own cluster, and by download this database, download the STAR pipeline, analyze using your own cluster, or you can do it in the cloud. For example, using the Anvil or Biodata Catalyst, but in that, so it would be good for the student to learn how to write app, and also using the workflow language and WDL language. So right now, to share the functionally annotated data for the whole database and the Favor backend database, and this is publicly available and in this online portal, and also Favor annotator can also be downloaded from the Favor website. And also for the TopMed and the CCDG data, and the annotated TopMed data are available in the Biodata Catalyst, which is NHLBI data common. And then for the CCDG data, we have just finished the QC of the FREE3 that include 140,000 whole genome, and then we just uploaded the data in Anvil, so people can apply for access and to the TopMed data using the dbGaP, and for CCDG data, it will be available to the community sometime probably later this year or sometime early next year. Okay, so let me do a little demo, and to show you how the Favor look like. Suppose I want to annotate, you can go to this website, favorgenohub.org. Suppose we want to annotate RS7412, and this is a very well-known causal variance in ApoE, and so you can see it take less than a second. You can, so because this whole database has 9 billion variants, because this is relational database, and so therefore the query is very, very quick. And so if you look at this, you can see that so this variance is a pass variance in TopMed. It provides allele frequency from the TopMed and NOMAD and 1,000 genome, and also provides a ClinVar information. This is a drug response. This has a drug response, and also particular phenotype. This is pathogenic variance, and so this related, we know this is related to cholesterol. And also this is this variance is exonic variance, and also this is non-synonymous variance. And also it provides overall allele frequencies in both TopMat, NOMAD, and 1000 genome. It also provide ethnicity-specific allele frequency in 1000 genome and also in NOMAD. And so basically, this is one step, and you can get all the informations. And also, it provides integrative scores, and I'll talk about this APC annotation pieces. So you can see this one has, because this is non-synonymous variance, and the APC protein function score is very high, the 37 is in the top 0.02% in the genome. So this is very functional. And also it has a good conservation score, it's in the top 3 percentile, and also has doesn't have a regulatory function. And so it also provides the detailed protein functional scores and also detailed conservation scores, and also provide epigenetic scores from the ENCODE as well, so that K27AC score. Those are not tissue specific. And also, so if you want to annotate a gene, and for example, if I put in ApoE, and so you can do the gene level query. And so this all shows all the variants observed in TopMat. So like in freeze, this will be freeze 8. And so right now for freeze 8, so ApoE TopMat observed about 14,000 variants and in ApoE, and so you can see about less than 1% are common variants, and so about 44% are singletons, 18% doubletons in ApoE, and also among those 1,400 variants, and then about 24% are exonic variants, and 10% are UTLs, and also provide introns and upstream, downstream. And also among those 1,400 variants, and 3 variants are drug response variants observed in TopMat, and then about 10 of them are pathogenic variants observed in TopMat, and then also provide about 8 of them are the loss functional variants observed, and also about 200 of them are non-synonymous variants, and so on. And so, and also one can do the batch annotation. Suppose you have the GWAS study, you have the top 1,000 variants of your favorite. You can upload your 1,000 variants positions or S numbers that will provide annotation of them. And also, if you have the whole genome sequencing data from the UK Biobank, for example, you can download the whole database, and then that can be used to annotate any of your whole genome sequencing study. Okay, now let me talk about how to perform the real variance association analysis. And so this using the STAR method. And so this paper was first published in Nature Genetics in 2000. And also this STAR pipeline, and basically is a follow-up paper that allow automatically annotate the whole genome and perform more advanced analysis that will appear in Nature method. So the main idea here is to boost the power of the real variance association test by dynamically upweight the functional variance using multidimensional functional annotations. So first of all, when we have a whole genome sequencing study, what analysis can we do? And so the input will include the phenotype and the covariates such as annotation PCs, and also because many of the samples are related samples, we need to construct the GRM. And so because the data are large, and to make it scalable, and we use the sparse GRM instead of the dense GRM. And so then we have the genotype data, and then use the favor to annotate the genotype, whole genome sequencing data, create this AGDS file. And the compression rate is 1,000 times, so therefore it's much smaller compared to VCF file. VCF file is just too expensive to store, and this can be easily read into R for analysis. And for the common variants, one can perform single SNP analysis, and for real variants, one can perform 2 types of analysis. One is a gene-centric analysis by creating the gene-specific mask, including the coding mask and non-coding mask. For genetic region analysis, one can perform either the fixed window a fixed size sliding window or dynamic size sliding window. And so now, what kind of mask can you create? So remember, when we analyze the real variance, and it needs to be grouped into variance set. And for the coding variance, the variance can be grouped into the loss functional variance and or missense, disruptive missense variance, or the synonymous variance. And for non-coding variance, they can be grouped into the promoter enhancers and also the UTL and so on. And this is where the challenge and art is, because the enhancer and promoter are cell-specific. And also, because the gene-centric analysis only covered probably 30% of the genome, and so in order to cover the whole genome, and because many intergenic regions, one can use the sliding window and with the fixed window size or dynamic window size. So because there are many annotations, for example, like ENCODE has over 30 annotations. And then how can we do the dimension reduction to annotate the variance? And so here we calculate, we call the annotation PC. This is different from the ancestry PCs. Ancestry PCs are calculated for each person. Annotation PC are calculated for each variance to measure the functionality. So here create the heat map. So if you can see that this big block basically are the heat map of the 30 epigenetic score from ENCODE. And so this red spot here are the protein function, about 10 protein functional score. And so this block here is about 10 conservation score. So what this figure tells us is there's not much correlation between the conservation and the epigenetic score. And so what that tells us is that in order to measure the functional information, function of the variance, we cannot say a variance is functional or not. We need to use multi-dimensional measure to measure the functionality. And so therefore, we create these annotation PCs. So for example, the APC, epigenetics, basically is the first principal component of the 30 epigenetic scores and in ENCODE that will measure the epigenetic function. And the APC conservation will be the first principal component of the 10 conservation score that measure the conservation of the variance and so on. Now, after those are all available and in favor, and now suppose you have the top metadata, how can you perform the analysis? And so the input data basically is the genotype data, and because many of the samples are related samples, and so therefore we create a sparse GRM to measure the relatedness, and so basically, you use a PC to control for population structure, use the sparse GRM to account for relatedness. And then we have the phenotype and the covariates. And now we annotate the genome under the top metadata. And so running the favor, so basically suppose we have this a billion variants, and then we have those annotations, hundreds of annotations, and to annotate them. And then from the by running the favor, that will provide you the annotations, calculate the APCs of epigenetics of each variance, APC conservation of each variance, APC protein function of each variance, and so on. It also creates a mask that can be used for analysis. For each, for example, for gene-centric analysis, that will create all the groups, the loss functional variance together and or missense variance together, or it creates a sliding window. Now we need to perform association analysis for each mask. So for example, we can run the SCAT method. I'll explain what that is. And then using a different functional annotation as a weight. And we can use APC epigenetics as a weight, calculate the SCAT p-value, or APC protein function as a way to calculate the scattered p-value, and then we aggregate them using the ACAT method. Or you can use a burden and with a different weight and then calculate using the scat to combine them. And afterwards, you combine them all using the ACAT method. Again, this will provide the robust star p-value. And then basically for each mask, by the end, you have a p-value that tells you the association between the promoter of enhancer and LDL. So then how can we do this test? So basically, suppose you have this marginal association statistic, and basically we use the score statistic because computationally it's much faster. We just need to fit the null model once. And then so you got this, suppose we have 100 variants in the promoter region, so ApoE. And then this will be your score statistic. We need to aggregate those 100 score statistics. And then one way to aggregate them is just doing the linear combination. This is called the Bowden method. And this W is the weight. And so you can see this will be powerful if all the variants are the effect on LDL are in the same direction. Then when does this work? Generally, if the variants are loss function variants, generally the effect are in the same direction, Bowden will work well. However, if the variants are in different directions, and in that situation, the Bowden will not work well because some beta are positive, some beta are negative, and if you use a linear combination of the association statistic, the effect will be cancelled out. So that's why you want to square it. And so this is basically the scat method. And so this will be powerful if the effects are in different directions or some of them are null variants. And so when does this happen? Generally for missense variants or promoter and enhancer, the effects are more likely to be in different directions. In that situation, Bowden generally does not work well. And now, because we use a different annotation as a weight, and so we need to combine them. And so what is a good way to combine the different correlated p-values? This is where the ACAT method come in. This is quite a powerful method to combine correlated p-values. And so suppose we have 100 p-value for 100 SNP. And then because the SNP are in LD, how can we combine them? The p-values, we can use the ACAT method and to do this transformation of the p-value to Cauchy distribution and then combine them. And so the ACAT method can be used to combine the SNP level p-value within a mask to calculate SNP set level p-value, or it can combine the p-value of different SNP set, and so then because use a different weight, and so then that can also help improve the power as well. So basically, suppose you use the ApoE promoters, and then suppose we use allele frequency as a weight, we upweight the real variance, downweight the common variance, and then we calculate the scat p-values, or we can use the CAD as a weight and to calculate the scat p-value, or use APC epigenetics as a way to calculate the scat p-value, or use APC conservation as a way to calculate the scat p-value. Because for any particular mask, and so some functional annotation may work well for one mask, but not for the other. So we want to create a robust way to calculate the association test. So that's why we use the ACAT method to combine all those p-values and using the different weight, because all those tests are calculated from the same mask. They are correlated, and by using the ACAT, that can handle this correlation beautifully and can be very quick. It takes only less a second. So computationally, it's very scalable, and because we make every step we can think about, make it scalable. So for example, for the lipid top-mat data for the gene-centric analysis only take half an hour, and for UK biobank data will take 1 hour. And for gene-centric genetic region analysis, and for the top-mat 30,000 samples, about 15 hours, and for UK biobank data is estimated using a day. And so here's some result on using the 30,000 lipid top-mat data. And so you can see that for the loss functional variants, PCSK9, this is well-known gene which already has a drug developed, and so the loss functional variance is significantly associated with LDL, and also like NPC1L1, there's already drug and target was this gene, and the missense variance of NPC1L1 is significantly associated with LDL, and so on. So one thing which you can see that is the promoter and enhancers of the genes and the effect generally are weaker. So then a natural question to ask is how can we improve the association in the enhancer and promoter regions? And so this is where the omic data comes in. So if we know for non-coding variants and the functions are tissue and cell specific. So if you look at this example, if we use for the lipid, and the liver will be the relevant tissue. And so if you use the liver as the weight annotation, and you can see that you have more discovery, that is the red curve here, compared to using the heart or the brain. And we know that the relevant cell type for LDLs are hepatocyte, adipocyte, and so on. So can we do something better using the cell-specific annotation instead of tissue-specific annotation? And so that is where the single cell data come in. And so we are currently working on developing a cell star so that improve the rare variance association test using the single cell based, single cell specific annotation. And so what are the challenges? And so using the single cell data, integrating them with the whole genome sequencing data. The problem is many of the single cell data available right now are the RNA-seq data. And they basically provide the expression profiles. And so, for example, those available in the human cell atlas. The problem is we need more than the RNA-seq single cell level data. So we need the single cell variance regulatory function and the regulatory elements, and also the mapping. And because the regulatory element map to the genes, and those are cell specific, those are very challenging right now. So basically, how can one identify the cell-specific enhancer for ApoE, and also for any regulatory elements, how can we map them to the gene at the cell level? And this is where the challenge and art is, and also that's where the limited data available. So right now, in order to do this, we need the single cell epigenomics data, such as a single cell ATAC-seq data and ChIP-seq data, and a single cell Hi-C data. And so that is why the NIGR launched this IGVF consortium and to help with this effort. Some preliminary data has been published. This is a very nice paper and by Bing Ren's group in the Catalyst, and they developed this Catalyst database which provide single cell ATAC-seq data about 200 cell types. The problem is that we don't have the data which both the single cell ATAC-seq data and RNA-seq data generated from the same sample. We do need those in order to do the mapping. And so this is where the field is needed right now. And hopefully the more such data will be generated by IGVF. So to wrap up, and so we provide a comprehensive whole genome sequencing variance functional annotation database and a portal for all 3 billion positions. And also, Favor Annotator can functionally annotate any whole genome, whole exome sequencing studies, and also by dynamically incorporate a multiple variance functional annotation. And the STAR can boost the power of the real variance association studies. And also tissue specific functional annotation can help improve the power of the real variance association test. And the STAR pipeline website is available here, and one can download it. So, this STAR pipeline paper will appear in the Nature Method. And so because in the interest of time, I want to have some time left for questions. So I will just mention what is the challenge for the meta-analysis. And so the major challenge for meta-analysis for the real variance association test compared to the common variance analysis is we need to store not only the marginal association test statistic, we also need to store the covariance, basically the LDs. And so because whole genome, like you think about TalkMed, has 1 billion variants, store LD is a huge, huge effort. And so how can we create efficient way to store the LDs and store the summary statistics, which include both association statistic and LD statistic? That is where this meta-star procedure is, and basically stores the summary statistic more efficiently and also incorporate functional annotation. And so this paper will appear in Nature Genetics. I think I will not go through the procedures, but I will leave some time for questions.